Greetings, my name is Grant Harrison and the date is June 27th, 2021. I'm here to demonstrate gene mapping for you all. So when you're mapping genes, the first thing you want to do is you want to determine the groupings. So you can see right here on the left-hand side of the data is the data set. And this data set gives us numerical values and the genes as well that are associated with those values. So right away when we look at these, we want to establish how the numbers may be grouped together by their similarities. So we can see that these two numbers right here right away are very close to each other, and they're very close to 1,300 thereabouts. So we can also, we'll bring that over and we'll take that highest grouping and we can establish that as being our non-crossover group. After we establish the non-crossover group of these two largest numbers, we want to look for the lowest numbers. So here we have 100, there we have 140, and we establish those, we group them down here on the bottom, and that's going to be our double crossover group. Okay, so we've established our non-crossover group, we've established our double crossover group, and now we're going to take those two remaining groups of two like numbers, and we're going to put them in the middle here, and those are going to be our two single crossover groups. And then another thing we've done here is we've taken the sum of each of these groups and we've just added them together because we'll need those numbers for further calculations. But here we have them designated as non-crossover, single crossover, double crossover with the associated values. The next thing we want to do is we want to determine the gene arrangement. When we determine the gene arrangements, we want to first look at one of these bottom numbers for the double crossover group. So we want to take the symbols of the values that were given for the double crossover group, and we want to try to essentially make a shape. So when we have a crossover, let's just use possibility number one. We're actually going to cross over these two genes with each other, and then we're going to cross over these two genes with each other. So A and B, and then B and C. So our job is to figure out what order they're in. So when we do that, we want to take these values right here, the A++, and we want to see if we can find A++ somewhere. Well, okay, so this is not, when you take this shape, what do we have here? We have B plus and plus. Okay, so that's not it. And then so here we have C plus and plus using that shape. Finally, we can see which one is the correct answer. We have A++, A++ gives us that shape. Now, if we were to use this other number or this other set here in the plus BC, we would still have that. We would have plus BC. It would just be the inverted shape of that. So it doesn't really matter which one you use as long as you're consistent with when you use it. Another thing we've done, or I've done, is I've actually rearranged this. So now that I found out that this is the gene arrangement is BAC, so finding that shape tells us we have BAC. Another thing I've done is I've changed these around. I've moved the B to the outside, the A to the middle. Same thing here, B to the outside, A to the middle, to give us that BAC arrangement. And that helps me just take better bookkeeping and kind of a way to double check what I've done. I've done B to A and then A to C. And what that also does is that helps me see that this distance right, or this number right here, that's going to help us get our distance from A to C. And this one right here is going to help us get our distance from B to A. So I have those listed out here as well. Next thing we want to do is calculate the distances. And so we use the numbers that we attained from before, and we're going to add all of those up, and we're going to get a total of 5,742. And so what we're wanting to do is we're going to want to find out what each of those numbers are on a percentage of the whole. So it's going to basically give us the total of 100%. But each of these divisions, so we're going to divide 2,702 by 5,742, and that's going to give us as a percent and a decimal. First, it's going to give us a decimal. We could convert that to percent if we wanted to, but we're going to take that. That's going to be our decimal form. that's going to help us get the distances. So when you're calculating the distance, now we have, so here you can see we have B to A to C. Ignore those values for now, but how you find that distance is you're going to add this 0.344 plus the double crossover groups. You're going to take each of the single crossover groups and add the double crossover group to it to get your total distance from A to C and from B to A. So you can see that these two numbers here, 0.142, 0.142, 0.041, 0.041, that's our total distance for one of our single crossover groups. That being from B to A is down here. From A to C is down here. So this is sort of the map of it. And so you can see below, I changed it from basically the percentages is what helps us kind of keep track of. This is not 0.183 map units, this is 18.3 map units. I've just moved the decimal place over 2 spots to give us map units. So from B to A is 18.3, from A to C is 38.5. Another thing we want to take note here lastly that will apply to this next slide especially is that we're looking at that double crossover group I have highlighted here in yellow. Well, that is going to be our observed value for the double crossover. So we're actually going to make further calculations using that number. And that calculation that we're going to be, one of those calculations we're going to be doing is calculating the coefficient of coincidence. So when we calculate C, the coefficient of coincidence, all we're doing is taking the observed divided by the expected. We don't have the expected yet, but it's very easy to obtain. Remember, our observed from the last slide is 0.041. And to calculate the double crossover expected, all we do is multiply the single crossover groups in decimal form from the previous slide. So we had our single crossover group distances being 0.183 multiplied by 0.385. That gives us a total, or a product I should say, of 0.070. Okay, so that's our expected. So observed over expected, 0.041 divided by 0.070 equals 0.582, and that gives us our coefficient of coincidence, C. We can take that, our value for C, to help us calculate I. What I is calculating interference. So for I, it's a very simple calculation. You just say I equals 1 minus C. So for this example, 1 minus 0.582 that we had calculated from before equals 0.418. Now in decimal form, that would translate to 41.8%. So how we interpret this information? Well, what that basically helps us do is formulate a sentence that, well, first of all, it's a positive number. So it may also be positive or it may be negative. So this number being positive means that there's 41.8% less double crossover than expected. A negative number would mean that there was more double crossover than expected. So one can easily establish the difference in examining the value for C. So before we had 0.041 is less than 0.070. That helps us formulate the sentence that the observed is 41.8% less than the expected. So that helps us make that calculation, and that is how we map out the gene arrangement as well as the distances, and that helps us gives us the coefficient for coincidence as well as the calculation for interference. Thank you. Well, in summary, in conclusion, here are the steps needed to solve this type of problem. First, determine the groupings. Second, determine the arrangements. Third, calculate the distances between the genes, signifying the observed double crossover value. And then lastly, calculate C and then calculate I. And that is all.